A very good evening to Dr. Gaurav Goyal sir and to the all respective audience of Pharma Lok. Dear audience, as you all know, Pharma Lok organize faculty and student development program on the occasion of World Pharmacist Day. That FSDP program have 10 series and this one is fourth lecture. I, Dr. Neha Tyagi, member of Pharmalok, on the behalf of whole Pharmalok team, I feel very privileged to welcome our eminent speaker, Dr. Gaurav Goyal sir. So before the starting session, I would like to introduce Dr. Gaurav Goyal sir. Sir, you are currently working in a position of Deputy General Manager and heading Transdermal Formulation and Development Department of Zydus Life Science and formerly Zydus Life Science known as Zydus Cadila Ahmedabad. Sir has total 15 years of industrial and academic research experience and worked with well-known industries such as Shilpa Medicine, Akums Drugs and Pharmaceuticals and Rusan Pharma Limited. Dr. Goyal had completed his doctorate and master's studies pharmacy from the Institute of Chennai Technology and graduation in pharmacy from Jamia Hamdard, New Delhi. So this is a very brief introduction, dear audience. If I would explain it takes so much time. So I invite our eminent speaker for their word of wisdom, and of course, I assure you, we would like so acknowledged. So, sir, I invite you.
Thank you very much. Thank you very much, Dr. Neha, for the uh, introduction. I'm very thankful to Mr. Amit Jha, who is the National Coordinator of Pharma Lok, to invite me on this Faculty Development Student Program on the occasion of World Pharmacist Day. There is just a very short correction in my introduction. I did my PhD and MPharm from the Institute of Chemical Technology, which was formerly UDCT, Mumbai.
Mumbai.
Uh, yeah, thank you very much. With this, I would like to start with my presentation. So today presentation is on the development of dissolution methods. Uh, can Pharmalok can start presenting the slides? Yeah, thank you very much. So today's topic for discussion is development of dissolution methods in consideration with the regulatory considerations, basically regulatory of US FDA I'm talking about, because these US FDA is generally taken as the uh, ballpark for all the companies, all the industries, and for harmonization, US FDA and European guidelines are generally followed. So, I start with this presentation. It will be basically for the students ki how to de- develop the dissolution method for your in your research work. It will be very helpful for the academic teachers also कि how research to be taken place to be carried out for the development of dissolution methods, how what care needs to be taken. Dissolution is not a new topic, everybody knows about it. I think the B farm in B farm we study about the dissolution apparatus, and we generally know the basket apparatus, pedal apparatus, USP 1 and 2 apparatus. But how what are the things how to interpret, that is we are going to discuss next today. Next, yeah. So the highlights is why we require the dissolution test and what is the acceptance criteria for dissolution test, and what are the desirable attributes for the dissolution test method development, how media to be selected, how RPM to be decided, time points for the dissolution method, how to derive the DR limits, dissolution release limits, and how to apply design of experimentation in development of dissolution method. Next. The dissolution method, why we require dissolution method? So generally when I ask this question to any of the students during the interview, or this is a very basic question when we ask why you develop the dissolution method, so they generally say to uh, make the method which is reproducible or which can be correspond to the in vivo method. Like if it is an oral once a day tablet or sustained release tablet, it is for 12 hours or 24 hours. The dissolution method should also be 24 hours. Or dissolution methods indicates how this tablet is going to behave in vivo in the body of the human being. So this is the answer which I generally got from the students or during the interview, but this is not the correct answer. This is uh, generally how the tablet is going to behave in vivo. In vivo means inside the body of the human being. We cannot predict through in vitro unless and until we develop the in vitro in vivo correlation. Suppose I want to say a tablet is completely releasing or dissolving 100% of the drug in 2 hours. It doesn't mean that in human being in vivo it is also going to dissolve in 2 hours the complete drug. Maybe it will be sustained release doses form which will be released in 24 hours, the 100% release. So then why dissolution? Then what is the then again the question comes, if this is not the right answer, then why we are doing dissolution? Dissolution, it is basically a test. It is a quality control test in which we are going to predict, we are going to actually find out whether that particular batch is is as per the quality specifications or not. So this is a batch release test because each and every batch before going to the market, it has to be released from the quality control department. So this is whatever dissolution limit or specifications we design, each batch should follow that particular specifications. So, like I just give you an example, if one batch is releasing 40% of the drug in 2 hours, but the next batch is releasing 60% of the drug in 2 hours, and the limit is 30 to 50%, our specification limit is 30 to 50%. So the first batch which is releasing 40%, it is within the specification, that is within 30 to 50%. But the next batch, it is releasing 60%. It is out of the specification. It is not within the specification range, that is of 30 to 50%. That means that batch is failing in the quality control test. So it is a quality attribute. It is a CQA. It is a critical quality attribute dissolution test, which is for the routine quality monitoring of the batches. So it doesn't mean a sustained release tablet, which is releasing for 24 hours in human being in vivo, can have the dissolution method of 4 hours or 2 hours. It can. It can. Then what is the then that we are going to study today? That this is an important point. So first of all, students, you have to bring in your mind, or the teachers which are present here, this is only the routine quality monitoring test, and there is certain benefits other than the quality routine quality monitoring of the batches. Like we can have the bio waiver during the application of our dosage form. Like we need not to do the bio studies. Bio studies means bioequivalence studies for certain drugs which are like basically for BCS class 1 and class 3 molecules that we are going to see in the next slide. And there is some SUPAC related changes. SUPAC means scale up post approval changes. Like after the approval of the formulation, but certain times during the scale up, we are need to change some formula or process. During that change, we have to show that the dissolution profile from the previous formula is similar to the new formula, the revised formula. So we have to introduce a dissolution profile comparison that it should be similar. So that is also we are going to study in the next slides. So it will be much more clear to you. Yeah, next please. Yeah. So what are the, like, if we want to have the biowaiver on the basis of the in vitro data, then we first of all we need to understand what is the BCS classification. I think the students which are there, just for your introduction, like there are four classes in the BCS. Class 1, which is highly soluble, highly permeable. Class 2, which is low soluble, high permeable. Class 3, which is high soluble, low permeable, and class 4 is low soluble and low permeable. So in this class 1 and class 3 are highly soluble drugs. Highly soluble means what? Certain times the students do not know the answer of this question also. So as from the industry, I just want to help you out. It is nothing related to the solubility in grams per ml. Highly soluble means the highest dose of any drug. Like for paracetamol, the highest dose is 1000 milligram. So 1000 milligram should be soluble in 250 ml, because the fluid capacity, generally average fluid capacity in human beings is 250 ml. So GIT tract, the complete fluid is 250 ml. So one highest dose should be soluble in 250 ml of buffer, and the buffer should be from 1.2 to 6.8. Means we have to, like, suppose in industries what we generally do, we take three buffers like 1.2, 4.5, and 6.8, and 250 ml in three different beakers, and dissolve the highest strength. Like for, like, cetirizine, it is 10 milligram. So 10 milligram we dissolved in all the three beakers: 1.2 pH buffer, pH 4.5 buffer, and pH 6.8 buffer. And then we see whether in this 3 pH it is dissolving or not. If it is dissolving completely in all the 3 pH, that means it is a highly soluble drug. If it is dissolving in only 1.2 and 4.5 and it is not dissolving in 6.8, then still it is not a highly soluble drug. Then we say it is a low soluble drug. So the highest dose should be soluble in 250 ml of all the three buffers. That means we have to cover the range from 1.2 to 6.8. Then only we can say it is highly soluble drug. So generally, the biowaiver we can receive, we need not to perform the bioequivalence if it is belonging to BCS class 1 or BCS class 3 molecules. But the exclusion is 2 and 4. If it is low soluble, then we have to do the bioequivalence studies, right? Then test and reference product should be identical. Identical means they have should be have the same sort of similar dosage form. It should not be like this. It is one is sublingual tablet and another is the immediate release tablet. It has to be identical. Their shape should be identical. Their size should be identical, right? Different salt forms we can introduce in the waiver also, right? And it is only for BCS class 1 molecule, and prodrugs can be also considered under the biowaiver. Whereas narrow therapeutic index drugs, which are very having the very narrow therapeutic index, means what it is can be toxic if it goes above the therapeutic range. So it is narrow therapeutic drugs we cannot include into the biowaiver, and in fact, the esters, ether, isomers forms cannot be included, and buccal and sublingual absorption drugs are not included in the biowaiver. Next, right. So this is a very important slide for all the students and all the viewers which are there. How to, because this is a very important thing which we generally during the studies should tell our students about the acceptance criteria of immediate release dosage form, because this is a general practice in industries, and students should, like, during the interview they should be having the knowledge of S1, S2, S3 criteria and L1, L2, L3 criteria. So S1, S2, S3 criteria, it is applicable for the immediate release dosage forms. Immediate release dosage form means which release the drug immediately. It is a conventional release dosage form. Generally, like in within 15 or 30 minutes, they generally release 80% of the drug. So now, if you see there, I have marked a test which is saying dissolution test where the specification is mentioned each unit dissolves 80% of drug substance in 15 minutes. Now 80% in bracket, if you see, I have written Q. So it is known as Q, means it should release 80% of drug substance in 15 minutes. Time point is 15 minutes. So what is S1 criteria says in S1, like we generally S1 criteria drugs is best drugs like which follow the S1 criteria. So we test 6 units, 6 tablets in dissolution apparatus. And what is the acceptance criteria? Each unit should not be less than Q plus 5. That means what? Our Q is 80. So each unit should be Q plus 5. 80 plus 5, 85%. So every unit should release 85% in 15 minutes. So if we see S1 criteria, there is a table which I have mentioned here for your better understanding. Like Q plus 5 is 85%, and we have tested 6 tablets, and the result are 87, which is above 85, that is good. 79, which is not complying. Then 87, which is complying. 80, it is not complying. 78, it is not complying. 87, it is not complying. That means 3 unit out of 6, it is not complying 85% criteria. That means this tablet is failing under the S1 criteria. So still, but we cannot reject this particular batch in industry if it is not for passing under the S1 criteria. Then we still have the provision as per pharmacopoeia. Then we can do S2 or S3 testing. So then in S2 criteria, we have to do additional 6 tablets, 6 units testing. Total earlier we had did 6 units, and now we repeat with new 6 units. So earlier 6 unit results is already there, which is similar in with S1 that we have to capture from S1, and then new 6 units we have captured. Now this units is now, if you see, it is 78, 79, 87, 83, 84, 82. Now what is S2 criteria says the average of 12 units, that is S1 and S2, should be equal to more than Q. More than Q means our Q is 80%. That means our average of 12 units should be above Q, that is 80%. So our average is here, it is 81.8%. That means it is passing under the S2 criteria. But there is one more clause. No unit should be less than Q minus 15. Q is 80, minus 15 is 65. So all units, all 12 units should be above 65. So yes, it is above 65 because our minimum is 76. So that means our all the units are above 65%, and as well as there is average is above Q, that is above 80%. So it is passing under S2 criteria. So we can definitely can release this batch into the market. So it is not only S1 criteria. If it is not complying under S2 criteria, there is one more clause, that is S3 criteria, where we have to do additional 12 units. So total means 6 plus 6, 12, 12 plus 12, 24. To 24 tablets, the average should be above Q, and no unit should be less than Q minus 25, and 2 units are not more than 2 units are less than Q minus 15. Q minus 15, not more than 2 units can go below 65% as per this Q criteria. So I hope this will be must be clear to you. You go through the pharmacopoeia. It is mentioned in Indian pharmacopoeia also, as well as in all the other pharmacopoeias. It is a very harmonized uh, chapter on dissolution and the specifications. Next, please. So this is on the immediate release dosage form. If we say about extended release dosage form, the criteria generally in extended release dosage form, the specifications are not one in it because it has to release. It is generally given in the form of ranges. Like here, I have given one example where the time point and specifications are mentioned on the right hand side. That is 1 hour, it is 20 to 40%, 6 hour 45 to 65%, and 12 hours it is not less than 80%. So if it is not complying, so what is L1 criteria? L1, L2, L3 criteria, it is for the extended release dosage form, where it is mentioned that No individual value should be less outside the stated range. Means all the six tablets, it is also done on six tablets, all should be within the range 20 to 40, 45 to 65, and the six units should not be less than 80%. If it is not complying, suppose one tablet in it getting 19%, 19 or 18%, it is out of specification. That is below range. The lower limit and the upper limit. Range is always having lower limit and the upper limit. So it is below the lower limit, 18%. Then we have to do again six tablets. So in the again six tablets, there we see average. So what is the average? The average of the 12 units should be within this range. First of all, the average should comply and it should be below the fall between the specifications. And what is the another criteria? Within the each of the stated range and is not less than the stated amount of the final test time. None is more than 10% of the labeled content outside each of the stated ranges. Means what? 20% का 10% कितना होगा? 2%. तो इसकी लोअर रेंज क्या बन जाएगी? 18%. तो हमारा 19 आया था तो वो इसके अंदर कंप्लाई हो गया. और अपर रेंज का 40 का 10% कितना हो जाएगा? 44%. 4%. दैट मीन्स तो रेंज बन जाएगी 18 टू 44. इज इट क्लियर? तो इट इज this type we have to see L1, L2, L3 criteria in this respect. I have not given the example. You can just go through the pharmacopoeia to understand this better. Next, please. So now we come to the development of dissolution test. This is the acceptance criteria, how to understand the acceptance of this batch, how to release the batch, and then now we are going to understand what are the desirable attributes of the dissolution test. हमारा मेथड कैसा होना चाहिए, क्या उसमें अच्छा होना चाहिए, like, right? तो a dissolution method should be discriminatory. Means what? Robust. Discriminatory मतलब हमारा these are the terminology. Okay, I am just reading out. We will go through slide by slide. These are in the next slide. Our method should be discriminatory, robust, correlated with the in vivo performance. Means how it is going to behave. Like it should be correlated. Like it should be having the correlation with the in vitro and in vivo correlation should be there. It should be transferable and reproducible. Means what? Like certain times with the same batch we are getting in 15 minutes 80% release, but again we are testing in some other laboratory with the same method. In 15 minutes we are getting only 40% release, but half of that. That means this method is not reproducible from lab to lab. In one lab we are getting some other results from the same batch, but in the another batch it is different results. So it is that method is not transferable or reproducible. That means shows that it should be having the control variability. Means the variability should not be like inter-unit variability. Like in the previous slides, we have seen the results of 6 units, and we have seen that the lowest most is 76% and the highest most is 87%. Right? So that means the variability from the average what we are talking about, that means it should be less than 20% at 10 minutes or less, or less than 10% at the later points. That we are talking about the average. So that means all the units should be having the low variability. That is RSD, what we are talking about. Complete release. That means 85% or is input. Every method should have be released. That means almost 85%, more than 85% of the drug should be released. And should, because then otherwise if it 80% the complete we are not able to recover the drug from the tablet, that means your selection of the media or your selection of the method parameters are not okay. Then we have to revise the method, right? And should be free of significantly stability problems. Means what? You have to select a media where the drug is stable. The drug should not degrade inside the dissolution media. It should remain stable up to the up to the analysis. We are going to do the dissolution, then we are going to do the HPLC analysis or either UV analysis. So it should be stable into that particular media up to analysis time point. Next, please. Yeah. So here is a slide how to I can like what we understand by this discriminatory method. The resolution method should be discriminatory. Means what? We will be able to identify the good and bad tablets. Like one batch we have produced and we add super designate, which is there in the formula. But in another tablet batch, during production, one of the operator forgets to add super designant. That means that tablet is not going to disintegrate properly, right? So that tablet dissolution should be lower. Definitely should be lower. But if a method is designed in such a way that we are getting the drug release from both the good and bad tablets same, there is no difference, then that method is not good. Here I have shown that in the screen the good tablet is having the higher dissolution profiling than the bad tablets. In the good tablets is little bit faster than the bad tablets. That means this method can able to discriminate properly. For discrimination, we generally find out in industries there is a one formula which is two parameters is for there similarity factor which is known as F2. F2, that is a similarity factor, it should be between 50 to 100, and F1 is a dissimilarity factor, it should be between 1 to 15. Then only we can say that the two methods, the two tablets, good and bad tablets, are similar or not similar. If it is similar, if it is both are similar, then F2 should be 50 to 100. That means our, but here good and bad tablet is there, so definitely F2 should not be between 50 to 100. It should fail, it should be below 50, right? If it is below 50 F2, that means the method is able to discriminate between good and bad tablets. But if for with the good and bad tablets, if we are getting the similarity factor between 50 to 100, it immediately shows that the tablets is similar, is showing the similar profile, then this method is not acceptable. So definitely our similarity factor should fail if the tablet is good or bad. If we forgot to add this super designant, so in industry what we generally do, we generally by intentionally we do not add certain excipient and find out whether the resolution method is able to discriminate or not. Means what? The similarity factor should be fail, should fail because because one of the excipient we have forgot to add, then it should, we should not get the similar dissolution profile, it should definitely should fail, right? So this is the importance of discrimination. Yeah, next slide. Robustness. In the robustness means what? The method should be robust. Means like it should not be too sensitive. Like if we are changing, if we are like the buffer we are taking for the dissolution media 6.8, but if you are changing the buffer pH from 6.8 to 6.6 and 6.8 to 7, so we are immediately getting different release. Like this is a graph which is there on the screen with this only the change of zero point 0.2 buffer pH, zero point 0.2 pH buffer, we are getting lot of variation in the dissolution profile. So the method is not good method. The method should be robust, it should not be too sensitive. It should not be too sensitive with the change of analyst to analyst, that one analyst is doing every time the results are similar, but when we have given this method to another analyst, we are not able to get the reproducible results. Lab to lab changes, vendor differences, like we are changing the excipient, like we have purchased one excipient like cross carbamylosodium from Merck. Now we are taking not from Merck, from Evonik we are taking cross carbamylosodium, so our results are changing. It should not be, the method should not be so sensitive that it is giving the vendor differences, right? So during this we have to, that is one of the reasons when during development we take lots from different vendors also. We generally do analysis from analyst to analyst, and we generally change the operators also. One, if in a laboratory, in colleges generally we have two or three dissolution operators. So like if it is there is provision of two or three operators, then we should do on one apparatus first, and the same lot we have to test on the another apparatus and see whether there is a method robustness or not. Next slide. Yeah. So what is the differences like areas with most robustness issue is developing method on unfit. Why we are what is the reason for this? Because we generally develop on the unfit system, not meeting the sync condition. Degassing may be the problem, sampling issues, filter selection is an issue, cleaning or carryover is an issue, or maybe we have not written the complete procedure. The SOP we have given is what is whatever we SOP we have designed. It is not clear for the analyst to do the analysis. So these are the reasons where we generally fail during the robustness. Next slide. Like in the dissolution system, if you see what I have seen, we are using the unfit dissolution apparatus. Like in the first figure, if you can see, there is a paddle, but this is the destruction paddle. There the corners of the paddles are not sharp enough. And in the second figure, we have shown the big basket. The basket is compressed. It is not a complete cylindrical basket. It is torn off. It is compressed from the between. So such type of variables, if the system is not fit, we generally get the problem during this. We should not have this significant vibration wobbling during the dissolution, the during the paddle movement or the cylinder movement. This can also leads to the uh, variability in the results. So these components of a dissolution operator should be properly placed, and it should be like annually should be maintained, or the operator should maintain the dissolution operators properly so that the results can be reproducible. Next slide. Now these are the dissolution operators which I have mentioned here, like the basket apparatus, which is the USB 1 apparatus, then paddle over it is, then this is a reciprocating type of system, and then we have paddle over it is USB 5, which is used for transdermals. Then we have the cylinder which I have shown in the fifth figure, which is the cylinder method, that is USB 6 apparatus for the transdermal, and in below it is USB 7 apparatus which are there, like which is reciprocating type of apparatus, which is also used for the transdermal system, right? So here I from this slide I just want to mention, like the generally we the choice as per the pharmacopoeias also, it is USB 1 and USB 2 apparatus, but we can consider a USB 3 and a USB 4 apparatus where we require the greater volume, or we like USB 3 apparatus. What is the importance of USB 3 apparatus? We can change the method media, like we generally like test for 1 hour in pH 1.2. Now in the next 1 hour we can do so without taking the tablets out. Generally, like it is between the basket. In the basket, we can change the media very fast. So this is the beauty of apparatus 3. Whereas apparatus 4, it is a continuous system where higher volumes is required for dissolving the drug, like for the very poorly soluble drugs or from the nanomaterials or from the microspheres. Generally, USB 3 and 4 apparatus are used. Next, please. Yeah. So ideal dissolution media, how to design, what media we should take. That is always the point of consideration, ki which media we should select. The first criteria for the selection of the media, that it should meet the sink condition. That means the drug should be highly soluble. What is the meaning of sink condition, that we are going to see in the next slide. It should be simple preparation for the media preparation. The drug should be stable in the media up to 24 hours and more, and we should use the little bit of, as much as little we can add surfactant and alcohol. Ideally we should avoid using the surfactants and alcohol, but if it is required, it is a very low soluble drug, then we may need to add some surfactants and alcohol to maintain the sink condition. Because the one important point which I just told, that at least it should be in asymptote. Asymptote means more than 85% of the drug should be dissolved into that particular media, right? So generally for immediate release tablets we use acids, but for delayed release tablets which has to be released in the intestine or large intestine, we generally use a very neutral media, and for the modified drug release we generally use neutral solutions, right? So delayed release tablet, we generally typically use acid for 1 or 2 hours, and then we go with the neutral media, that is changeover of the media. Next, please. So sink condition means what? For the proper dissolution, media is needed, which is capable of dissolving 3x API. Like like in one tablet, if you say like if you are containing 500 milligram of paracetamol, so at least media should be selected which can dissolve 1500 milligram of paracetamol. The volume should be selected and it should be sink condition has to be maintained through either through the media change of pH and either through the volume. The volume should be so much high that it should dissolve at least 3 times of the quantity of API which is there in the unit dosage form. Like in a tablet, it is 500 milligram. Like I have given example. For cetirizine, it is 10 milligram. So at least 30 milligram of the API cetirizine should be dissolved into that particular dissolution media. Otherwise, if the API it becomes saturated, it can be overly discriminating. Like we are overly discriminating means we are getting the discrimination like F2 value below 50. So method should be discriminative. It should be good or bad. It is going to discriminate. So that is why we generally use a sync media. Otherwise, method itself going to show that it is overly discriminatory. Next slide. Next slide. This we have discussed media selection. Next slide. Media selection, like one of the two important points which I already discussed, that we need to avoid the surfactants. But generally, surfactants, if required, we should be it should be below 1%. But if it is above 1%, then we should use some we should be like it will be very difficult to handle more than 1.5% of the surfactant. So generally we should avoid surfactant concentration below 1%, and it should be stay within the pH of 1.5 1.1 to 7.5. After adding the surfactant, the pH should not be more than 7.5. It should not be below than 1.1 pH. Next slide. So these are the some thumb rules which are used in industry during the development. Like certain media cautions, like it should be having the no buffering capacity. Be careful with water, because why we should not use water as such. That is the point. Generally, water should be avoided, and we should use buffers, because water is not having the buffer capacity. Like if you add certain tablet or because tablet contains API, that is your drug substance, as well as excipients, so they can change the pH of the media. When they are going to change the pH of the media, then your media, then every time you can have the variability in the drug release, and you are not going to able to understand why we are getting the drug release or variability in the dissolution, because the pH of the media is changing because of the excipients which is there in the formulation, or because of the drug substance. No, go to the previous. Yeah, yeah. So that is why we generally should use buffer systems, right? And one very important thing, do not use SLS with potassium phosphate buffers. This is a very important point. Generally, in interview, it can be asked like SLS cannot be used with the potassium phosphate buffers. We generally use sodium phosphate with those. You just understand with this, the SLS means sodium lauryl sulfate. It is containing the sodium salt. So sodium salt should not be used with the potassium salt buffers. You just keep in mind like this. Sodium salt, sodium SLS, sodium lauryl sulfate, it is containing sodium. It should be used with the sodium phosphate buffers only. So just in this way, you just understand this thing, right? Next slide. Media degassing is very important if you are using the surfactants and if drug is susceptible with the oxidation, because in water or buffer generally we have the dissolved oxygen in the media, and if you want to deaerate or we have to degas, then what is the method? USP method says that we should heat the media for up to 41 to 45 degrees Celsius, and then vacuum degas through 0.45 filter or less. Like in the figure you can see, this is the vacuum degassing process. We generally use vacuum, and then in the there is a donor compartment. From the donor compartment there is a beaker, like I am saying, we fill the media in this, and through the filter we get the filtrate down through the vacuum. So that particular process we can use, that is a vacuum filtration. This is known as vacuum filtration. It has to be degassed through vacuum filtration. This is the USP method. So USP vacuum filtration method is the most acceptable method in the laboratories, QC and industries. There is another method which needs to be validated. These methods are available in pharmacopoeia. That is helium sparging. That is, we can sparge the helium so that oxygen can be removed from the media. Automatic degassing systems can be used, or superheating systems, but generally USP speaks about it, but this method should be validated. Yeah, all the three methods. And if you are using the USP vacuum filtration method, that doesn't require the validation. Next slide. Yeah, agitation rate is very important, because all this is a game of hydrodynamics. Because whenever hydrodynamics should not be very high and should not be very low, because during dissolution the first process is the disintegration. The tablet has to disintegrate, then after disintegration the granules which are produced within the bowl, that is a dissolution bowl basket, it has to be dissolved from that particular granules or disintegrants. Excuse me. Means what? Here we have to understand this. Hydrodynamics plays a very important role in disintegration of the tablets here, as well as the dissolution of the active or drug substance from those disintegrant particles, that is granules. So for that we need to give certain hydrodynamic force to this dosage form unit. Either we generally in basket or paddles. We in basket, as per the pharmacopoeia, we should the basically we should select the RPM or agitation rate between 50 to 100 RPM, and paddles we should between 50 to 75, and which are and not allowed outside 25 to 150. That is a very important point. It should not be below 25 and it should not be higher than 150. It has to be justified with your method, why we are going with 25 and 150. Otherwise the very general rule for baskets it is 50 to 100, and paddles it is 50 to 75. In USP3, which is a reciprocating type of operators, where there is a tablet unit dosage form in a basket, and it is going up and down. It is moving up and down. It is reciprocating movement. It is there, and then it is generally we say it is dips. So it should be 5 to 30 dips per minute. And USP4 it is it is continuous solution which is moving inside the apparatus. It should not be, the volume should not be higher than 2 ml per minute to 50 ml per minute. It should not be higher than 50 ml per minute, and the lowest most is 2 ml per minute. Next slide. So generally for IR tablets, we generally give 1 point, one time point, like I have given in this, like before that, 15 minutes and 85%. That is 80Q I have given. Here it is 85Q is mentioned. More than 85% release in less than 15 minutes. That means here Q is 85. Generally we can give 1 point or 2 points. And if we have to for IR tablets, we can give up to 60 minutes also. For extended release tablet, we have to give at least 3 time points. One is the early time point, where we generally have the dose dumping or potential dumping immediately the drug is released up to 30 to 40%. Then intermediate release up to 50 to 70% release. That time point we should give, and then the final time point where the drug releases more than 80%. So we have to give the three time points, which I have shown in the graph extended release when I was presenting the that particular slide. Acceptance criteria for extended release tablets. I have mentioned the three time points over there also. Next, please. These are the sinkers which are generally used for the floating type of tablets or capsules, unit dosage form, which are coming up. So we can use a sinker so that it should be reproducible. Next, please. Yeah, this is very important slide in case there is a coning problem, because coning is a problem where when the density of the excipient or the drug substance is very high, it settles down in the basket at the bottom, and even with the higher hydrodynamics like 100 rpm, it is not coming up because it is so denser. Like generally, like with the iron tablets, we got this type of problem. So we can use sinkers. Sinkers means like this is the if you see the second basket, there is a itself there is a cone. It is no cone, so that the powder should not accumulate at the bottom. The shape of the basket itself, it is conical, so the powder should not settle immediately below the bottom of the paddle. And it is helping because the hydrodynamics will help to move the powder uh, up, move the powder inside the basket. It should not be the coning problem can be removed. So for this, a special baskets are you can purchase from the vendors. Next slide. This cross linking, which is a pellicle formation, it is a general problem which we generally seen in the capsule shells, which is made up of gelatin. So generally, this capsule gelatin cells does not dissolve properly. So in such type of tablets, we can use cross-linking agents like pepsin or pancreatin can be can be used so that the cross-linking of the gelatin can be decreased, and the drug should be immediately released and dissolved. Otherwise, certain types this capsule does not dissolve and release the tablet for 1 hour also, and it is an immediate release type of those capsule shells. So in that we need to use some pepsin and creatine into the media. Next, please. How to decide the sampling location? If you see here, I have mentioned the hydrodynamics in paddle apparatus. The red and the yellow is the where the hydrodynamics is very far, very high, and these blue area and green zones where the hydrodynamics is very low. When there is a low, that means it is not a proper mixing. So generally, we have to take the sampling from such an area which is a truly representative, which is a where the mixing is proper. So yellow, there is a lot of high hydrodynamics. So proper mixing is there. That is why we should, in this yellow area, we should do the sampling. Our sampling port should come. It should not be very near to the paddle, and it should not be very near to the basket wall, because if you see the basket wall, it is still blue. So it should be in between this paddle and the basket wall. Yeah. So the importance for this particular why we generally, because in most of the pharmacopoeia you can read that you should sample in between the uh, this paddle as well as the basket wall. What is the reason? Because it is the hydrodynamics plays a very important role over there for the proper mixing. Yeah. Next slide. Next slide. Next slide. Hello. Hello. Yeah. Thank you. The filter selection is very important. The filtration stops the dissolution process. Uh, means filtration must be there, right? Because filtration, after the filtration, why we should do the filtration? Because like generally after sampling, the undissolved particles also comes into the sample, like the drug particles which are not dissolved also comes into the sample. And if you keep the samples as such, the undissolved particles are going to dissolve into that particular sample, and you will get a good results in terms of dissolution. Like if it is dissolved only 60%, but you will get is 80% release, because the undissolved particles up to analysis will dissolve that 20% of the additional undissolved drug will be dissolved into that particular sample itself. Therefore, immediately if you do the sampling, the first process you should do, you do the filtration, because the filtration will stop the dissolution process. Whatever is undissolved, it will be filtered out, and you in the filtrate you will not get the undissolved particles, undissolved drug. So that is one of the reasons कि we should do the filtration immediately, but we should use the validated filters. What does it mean by the validated filters? In three terminology, I can explain this: efficiency, leachability, and adsorbance. Efficiency means it does remove the undissolved drug. Means undissolved drug should be eliminated from the uh, filtrate or the samples. It will be efficient enough, because the pore size should not be so such much. It should not be highly porous, or the pore size should not be very large. We are getting a pore size, our undissolved particles also gets passed down, and it goes inside the filtrate. Second is the leachability. The filtrate, the particles from the filtrate should not leach inside the filtrate, and we get the one more peak in the HPLC because of the filtrate itself. And third one is the adsorbance. The dissolved drug should not be retained by the filter. It should not be adsorbed inside the filter paper. Yeah. Next slide. So these are the three important criteria. This is the USB 3 apparatus reciprocating type, which goes up and down. And just to show you this apparatus, I have included this particular slide. And the beauty of this apparatus, that you can have the variable media, like in the one run itself you can have the like pH1, pH3, in the another tube pH4, pH6. So in the one method itself you can have up to 10 pH different buffers you can test, right? Next slide. This is USB 4 apparatus, which is known as the flow through cells, which is a continuous flow inside the cell. And generally, for the poorly soluble drugs like BCS class 4 molecules and BCS class uh, 2 and 4 molecules, we use flow through cells. On nanoparticles, we can use this particular apparatus, microspheres, right, where the bigger media is required to maintain the sync condition. Yeah. Next slide. Yeah. So this is the DOE. This is the last slide of my presentation here, which I have shown here. Like you can like in the first case in the table 1, here we have optimized the SLS concentration in the media, like 0% without SLS means zero point 0.1, 0.2, 0.5% in three different buffers. Like in the A means zero point 0.1 buffer, 1.1 buffer, 4.5 buffer, and C means 6.8 buffer. And then we added in this all the three media different concentrations of the SLS. And with the SLS concentration, we are seeing this, the solubility of the drug is also increasing. And if we see, the maximum solubility of drug is in the pH 6.8, that is C, with the concentration of zero point 0.5, right? If we from the table number 1, so the proper concentration, the this is the as per the DOE, the zero point 0.5, it is going to showing the good solubility. The researchers has used 0.45%, as a optimized level of this particular surfactant level, and again tested with ABC. That is a different pH, 1.1, 4.5, and 6.8, and DEF is the rest, is with the 0.45 buffer, P 0.45 percentage of SLS. So D is reciprocate of 1.1 buffer and 0.45% SLS. E is 4.5 buffer and 0.45% of SLS, and F is 6.8 buffer plus 0.45% of SLS. And then by this OFAT, that is one variable at a time, he has designed and record the results. And here we can see the E is giving the very good release up to 89% in 45 minutes. So E is the optimized buffer. So that this type of DOE, design of experiments, we can include in our studies, which will be very helpful in this during presentation in the thesis also, to explain to others on what basis you have optimized the concentration of surfactant, as well as on what basis you have selected the buffer. So I hope with this presentation you have get the insights of the development of dissolution method. Thank you very much. Thank you very much, Pharma Lok, to give me the opportunity to present to the students as well as to the academics how to develop the dissolution methods as per the regulatory requirements. Thank you very much for your attention. Amit ji.